Please start the webinar.
Please start the webinar. Please start the webinar. Please start the webinar. Please start the webinar. Please start the webinar.
Welcome to the Jigyasa Atal Tinkering Lab webinar series. This series was launched during 15 June to 22nd of September 2020 with a focus to overcome the myths in common public, especially students, on COVID-19 with respect to drug discovery, clinical aspects, and technologies combating COVID-19. These webinars have attracted a large number of students across the country and allowed their direct interaction with the scientists to inculcate a culture of curiosity by asking questions. The webinar series has been restarted once again as Demystify Science Series to be a part of CSIR's and Niti Aayog's Azadi Ka Amrit Mahotsav initiative. The first talk in this webinar series was by Dr. Shekhar C. Mande, former DG CSIR on Molecules to Life. Second was by Dr. Anil Bharadwaj, Director, Physical Research Laboratory on the topic Indian Planetary Mission. Third, by Dr. Chintan Vaishnav, Mission Director, AIM, Niti Aayog on the topic Demystifying Innovation. Fourth talk was presented by Dr. Mailswamy Annamillai on the topic Science to Learn or to Change. And the fifth talk was delivered by Dr. Mukesh Shivaria, Senior Scientist from CSIR NPL on the topic Realization of SI Unit Meter, History of Measurement of Length. Today, under the Demystifying Science webinar series, we will have our sixth popular talk by Dr. Anshu Bharadwaj on the topic A Silent Pandemic Leading to Pre-antibiotic Era. Dr. Anshu is the principal scientist from CSIR, Institute of Microbial Technology, Chandigarh. She is the group leader and long-term fellow, CRI Paris, France, faculty at ACSIR Delhi, India, team lead, Drug Discovery Virtual Tool Room, Drug Discovery Hackathon 2020, an initiative by Government of India. Dr. Anshu Bharadwaj, a biologist and a data scientist, obtained her PhD in 2008 in understanding genotype-phenotype correlations in human mitochondrial diseases from CSIR CCMB, Hyderabad. She has over 40 publications to her credit and has trained over 500 young students. She served as an associate scientific advisor to Science Translational Medicine and is on the editorial board of Journal of Genetics, review editor of Frontiers in System Biology, Protein Bioinformatics and Computational Genomics, and advisor to F1000 Research Galaxy Gateway. She has several honors and awards to her credit. She was one of the 4 young innovators in India selected for the prestigious International Visitor Leadership Program by the US State Department. She was also awarded the Newton Bhabha Fund from the British Council and Royal Society of Chemistry, UK, and long-term group leader fellowship by CRI Paris. More recently, her lab has developed a unique scientific video game for the awareness on AMR, which is endorsed by WHO India. Her passion is for biomedical big data analytics, a platform with a focus on understanding infection and rare disease biology for better therapeutics and diagnostics. Today's talk is on the topic, A Silent Pandemic Leading to Pre-antibiotic Era. As you might be knowing, regarding the overusage of antibiotics during the COVID pandemic, WHO was compelled to send alerts to stop its global misuse. To make the kids understand what it all means, Dr. Anshu Bharadwaj has developed and programmed be able to play and learn about the antibiotics with the help of this game. Today's talk would highlight about the Nobel Prize winning discovery of antibiotics and medicines to cure infections. it is important for us to understand the concept of antibiotic resistance and also the consequences of irresponsible usage of antibiotics. Dr. Anshu Bharadwaj, over to you, ma'am.
Thank you, Shraddha, for that nice introduction. So let's get started. Can you see my screen?
Yes, ma'am.
All right. So I can hear myself back. So if there is anybody who is on unmute. Okay, good. So welcome all to this Demystifying Science series. So I'm really glad to be here today because the reason why we have developed a video game is to generate awareness on a healthcare issue that actually impacts all of us. And that is what reflected in the title of today's discussion is a silent pandemic leading to pre-antibiotic era. Now, as most of the audience today is expected to be young kids from school as well as general audience, I have kept the scientific jargons in this presentation to a bare minimum so that we collectively understand what the actual issue is and how we can go ahead and deal with it as a community. So let's start by at least understanding what the title means. So when you start to use, when we start to use the word pandemic, which is actually not a new word to anybody on earth today, but for better clarity of the young students, how do you define a pandemic? So pandemic has originated from a Greek word which called pan, which is all, and demos means people or crowd. And when an infectious disease actually spreads across a very large population in the world, you start calling it a pandemic. And we all actually are witnessing that in the COVID-19 pandemic. Now, antimicrobial resistance, which we'll come to a little later, is a silent pandemic. But how is it that leading to a pre-antibiotic era? We all know what antibiotics are. They are basically medicines that we take to prevent infection because they allow us to kill bacteria, right? Now, they are basically very heavily used. All of us have taken antibiotics at some point. So when I say it's a pre-antibiotic era, what we are talking about here? So pre-antibiotic era equates to the fact that when antibiotics are not working anymore. See, antibiotics helped us to prevent and control communicable diseases. And if these antibiotics stop functioning, then even common infections and minor injuries, which have been treatable, may actually start killing millions again. So this translates to the fact that in the pre-antibiotic era, when we had no antibiotics, we are actually in the same situation right now, at least for a few drug-resistant bacteria, where antibiotics are not working anymore. So something which is not there or something which is not working means the same thing, right? And that phenomenon, when your antibiotic was working, but now it is not, it is called as antimicrobial resistance. Now, is this something new? Actually not, because Sir Alexander Fleming, when he discovered the first naturally occurring antibiotic, and we all know penicillin, in his Nobel Prize winning speech, he very clearly said that it is very easy to basically make bacteria resistant to penicillin by not providing them sufficient concentrations to kill the pathogen. So he also said that we, being ignorant, can do antibiotic misuse over a period of time and the antibiotics will stop working. Now that was something that he was proposing at that point in time because there were already reports of penicillin resistant. He could see that, so he made sure that all of us also understand that there is going to be a problem and therefore we need to be very judicious and rational in usage of antibiotics. But then what happened? We really were not careful. And if you look at the recent data, it actually communicates to the fact that what doesn't kill you makes you stronger, right? How do we know what he hypothesized way back in 1945 is true? If you look at the deaths caused by antimicrobial resistance in 2019, you would see that approximately 5 million people have died from illnesses which are somewhere related to AMR. And if you can see on the slide, there are certain pathogens like E. coli, Staphylococcus aureus, Klebsiella. So they cause infections in a very large number of body parts, and they actually can lead to mortality. It is not an infection that is not killing now, it's an infection that is killing. Why I am showing you a data which is 2019? Because due to the COVID-19 pandemic, we don't even know the damage, the actual intensity of the damage caused by increasing use of antibiotics in COVID-19 pandemic. But even before the pandemic, what estimates indicated is that if we are not addressing the problem of antimicrobial resistance now, in next 25 years, we are going to have a situation where one person can die of resistant bacteria every 3 seconds. So it's going to be very, very difficult to handle this situation if we don't do anything right now. Okay. And that is why understanding what goes wrong and how do we control our behavior becomes very important. So what normally happens in a population is, if you see here on the slide, what you see are bacteria which are silver colored and the ones which are in golden color. The silver color bacteria are the ones which we called as susceptible, which means they can be killed by antibiotics. The ones which are in that separate and a golden color are ones which are resistant to antibiotics, which means that somehow they have identified a mechanism that antibiotics have stopped working on them. So what happens when you give antibiotics? The ones which are susceptible all get killed. Now there is enough food and nutrition for the drug-resistant bacteria to survive. So by giving antibiotics, we are actually selecting or either basically allowing the drug-resistant bacteria to proliferate. And this is a very dangerous situation. Now, how does this happen? So a susceptible pathogen or a microbe, when there is no antibiotic, obviously it will grow happily. But when you apply antibiotics, its growth gets inhibited because the bacteria dies. But when a microbe becomes resistant, even in presence of antibiotics, it will behave as if there is no antibiotic present. So how does it do that? So there are actually so many mechanisms by which bacteria actually can do this. Some of the main ones are that it will not let the antibiotic enter inside the bacterial cell. So it is very easy, the antibiotic is not even entering the cell. How will it work? Even if the antibiotic somehow penetrates inside the cell, it will identify, I don't want this, and it will throw it out. So the antibiotics are pumped out. If it cannot do any of these two things, then it will try and degrade the antibiotics are nothing but chemical compounds, right? So then it actually, the bacteria make enzymes that then degrade these antibiotics. And other mechanisms also where it will not let the antibiotic function. Now you will ask a question, how will a bacteria know what to do in case of which antibiotic? This looks like they have some sort of training. So actually that is true. Reason is antimicrobial resistance is an evolutionary phenomenon. Now when you see these mechanisms in bacteria have not happened because of antibiotics. Antibiotics it's not even 100-year-old, right? In 1940s is when we discovered penicillin. But antimicrobial resistance has been there for more than 1 billion years. And the reason is there was a chemical warfare going on between different bacterial species. They all want to stay in the same environment. So they try and make antibiotics to kill the other one. Now, if the other bacteria has to survive, it will have to develop and evolve mechanisms of resistance against such attacks. So since they have been doing this arms race, this learning is going on for 1 billion years. So they are pretty evolved. So they know when an antibiotic comes or when they are basically under some kind of pressure to be killed, they know how to evolve, mutate and turn into something which is very difficult to then get rid of. Now the point is, you may very easily say that this is a problem with the pathogen. So what is that we can do about it? What is our fault? So the problem is, how are we responsible for antibiotic resistance? Is actually in many ways. I have listed a few common ones here. First is then we over prescribe antibiotics even when it is not supposed to be given. Say for example, in viral infections, antibiotics don't work on viruses, but we still prescribe antibiotics in viral infection cases. Patients don't finish treatment on time. If say for example, if you're given a 5-day antibiotic dose and after 3 days you feel okay, it's all right, and then you decide, okay, I'm not taking antibiotics anymore, those are very dangerous situations. This becomes even more evident in case of patients like tuberculosis because they have to take antibiotics for almost 6 to 8 months, and it becomes very difficult for them to comply to that treatment. And that is why, patients at times find it difficult to finish treatment. We use a lot of antibiotics in livestock and fish farming, obviously to fight infections, but also as growth promoters, because you need a fat fish or a fat chicken so you can sell it more, so you can make more profit. And that is why antibiotics are overused also as growth promoters in poultry and other industries. Then, of course, lack of hygiene, poor sanitation, poor infection control measures in hospitals and clinics. Because if there is more infection, then you will have to give more antibiotics, there will be more exposure. So one thing is for sure that if we can somehow prevent infections, we'll be able to also use less antibiotics. Now, one of the last things that you see here, lack of new antibiotics being developed. So one other thing that normally students will ask is that what is the problem? We have discovered so many antibiotics, why not one more? The problem is that discovery of new antibiotics is extremely difficult, both in terms of cost and time. To discover a new antibiotic and make sure it is good and safe for clinical use for human, it takes approximately somewhere between 10 to 15 years. And the cost you can see for a single antibiotic, it can take anywhere from $500 million US to 1,000, almost $1 billion US dollars. So the cost and the time investment is so high that if we will not learn to judiciously use antibiotics that we have now, that we know work, we are already running out of options. And this is something which will impact all of us, not just doctors or people who are sick, because in COVID-19 pandemic, we saw how something that, takes you by surprise can, the extent of damage it can cause. So we need to be prepared. Now, it's not that AMR is not discussed. It is discussed on scientific forums. It is also discussed in popular media a lot. But somehow, none of these actually have contributed extensively in behavior change in the wider community, like us. How do I change my behavior in terms of not pushing a clinician saying, okay, prescribe me antibiotics, when I'm not sure I should be getting one? So we are actually not in a position to take those decisions, and we thought it might be because we don't understand the problem. But bacteria does understand the problem. They have understood that we are abusing antibiotics, we are overusing antibiotics, and they are evolving. So we need to be smarter than, if we need to survive, then we have to learn to work out an ecosystem, an equilibrium in which we can survive with the microbes in a way that it's not that evolution is going to stop at some point. So we need to understand that evolution will keep on happening, and we have to be intelligent enough to be a part of that evolutionary process and learn. Bacteria is learning, they are mutating, and they are becoming stronger. So now the point is, because since it's such a complex phenomenon, and you will see why I'm saying that, what do we do in order for better engagement and better understanding of the problem in the very first place? So what we decided is, why not actually make ambassadors of change? And I think the ambassadors of change can be the citizens of tomorrow, young children like you, because what you can do, adults can never achieve. You can influence community members, you can influence your families, you can influence your neighborhood. And that is why we decided of investing a lot of energy and time in developing a video game, which we call as Armor. The game is on, and the reason we are saying the game is on, because it's an evolutionary arms race again. This is not something that has stopped, it's going on. And that is why we wanted to show that the armor is not just for the bacteria, but for the host human, for us also. So Armor is basically designed as an awareness game to trigger scientific curiosity and awareness on the issue of antimicrobial resistance. and to convey the message that if antibiotics are misused, underused, overused, it actually leads to accelerated resistance. And that you will see when you play the game. And we use certain technologies. I'm not sure how many of you are gamers there, but if any one of you would like to develop games with us, then there are a lot of technologies that are available for free to use. Like we developed this game in Unity 3D and C#. And we are also now developing a conversational artificial intelligence bot to interact with the players. This is something which is ongoing. So let's come back to the game. So there were a lot of options for us to design armor. There were like card games, there were card games, there were games where you can actually do pattern matching. There were a lot of options available. But the problem is that AMR, given a very complex phenomenon, we wanted something that can capture the complexity. So what we did, we went ahead with a game genre which is called as tower defense. And I'm sure most of you who play games will know what I'm talking about, something like Plants vs. Zombies. So in a tower defense game, what happens? So say for example, this is the mark that you see here with 3 hearts. This is basically the target that the player is defending. You don't want anything to touch your target. And what you have are enemies, which are shown here as smileys. So these enemies will follow a path and try to destroy your target. And the objective is to stop them doing it. All right. So what you do is you plant towers, and these towers will launch attacks on these enemies and kill them. But in case some of the enemies escape your towers and get, and they escape and they reach the target, then you lose life. So it's a very simple mechanism wise, and then the game starts again because you still have 1 life left. So what we have done here is we adopted tower defense to represent AMR. How did we do that? So the enemies here that you see here are the pathogenic bacteria. Towers are antibiotic dispensers. The arrows that you see here, which are bullets, are antibiotic bullets. The terrain type, or what you are defending the environment, is any tissue, like say for example skin infections or lung infections. And what you are defending is you are not letting the bacteria enter through any route in your bloodstream. That is what you are defending. The bacteria will enter your body, but you will now defend it by planting antibiotics so that bacteria does not reach blood capillary. Okay. And the story starts something like this. We wanted to give a context to the players. So what are they really doing here? So the story goes like this. There are 2 kids who are playing in a park, and while playing, they got hurt. Now they went to a doctor, and the doctor wants to exactly know if the wounds are infected or not. And once he checked that the wounds are infected, he is prescribing antibiotics, saying, okay, now you should take antibiotics. What happens after a week is that Alex's wound didn't heal. So one of the kids got healed, the other one did not. And now the game is about how do you help Alex heal his wound. That is the game about, and that is when you know what goes wrong when the wound is not healing and what are the problems that it is having. It might be a drug-resistant pathogen and the antibiotic is not working. Or maybe Alex is not finishing the course of antibiotic, or maybe he's taking too much antibiotics. How do we know that? Now, the reason for selecting a skin wound to explain the phenomenon is because wound is something that we can see. If I say lung infection or stomach infection, it becomes a little difficult to imagine that. So a wound infection is something that you can see because wound is something that you can visualize. And if you look at, for some of you, you might be seeing in your textbook that a skin, when you do a cross section of a skin, it looks something like this. So you can see these are your skin here. Then there is epidermis, dermis, and hypodermis. And somewhere here is what you see are your capillaries, the red and the blue ones, right? Now, the logic that we have applied is we have translated or gamified human skin, and bacteria through the wound will enter and will try and enter the bloodstream. Now, as a player, it is your job to now make sure that bacteria cannot come from the skin to the blood. And that this is where you will plant antibiotics. So let us see how the interface of the game looks like. So if you see here, it's a typical game interface. You have the number of lives, you have menu items and many other things. So what you see on this side of the screen is how a wounded tissue or the wounded skin looks like. And this is where from where the bacteria will enter. Now what you need to do is they will enter here and they will take this path and they will try and enter the bloodstream, which is here, and you see RBCs moving on this side of the panel. These are the places where you can plant your antibiotics, which you see here. And if you see antibiotics you need to buy them. So you need to have money to buy these antibiotics. So let me just play this for you so that you see it moving. So what you do is you take the antibiotic from here, plant them here, and then you start killing the bacteria. Once you start killing the pathogen, this particular bar goes up, and once it finishes, you have cleaned the infection. That means there is no more infection and Alex is cured. And that is when you go to the next level. Now, in the next level, what we want to convey to the players is that bacteria divide. So please don't wait for your infection to go bad, because bacteria divide very, very quickly. And if they divide enough, then there is absolutely no way you are winning the game level, because they will just overwhelm the system so much that you won't be able to win the game level. And this level, you will actually see a lot of bacteria dividing. Now, and one of the other levels, what you will see that this particular antibiotic that we were using so far, this tablet has stopped working. And even if you plant this, nothing happens. And if you see, the bacteria has changed its form, its color, there is some kind of an aura, a bubble around it. So it is now a drug-resistant pathogen. So it requires a different antibiotic, and that different antibiotic is shown here as a different tower. Now, if you see here, this antibiotic is more expensive than the previous one. And this is normally what happens as well, right? And sometimes what will happen is, despite the fact you have a new antibiotic, some pathogens will still escape the attack of the antibiotic, and they will still try and go and reach the bloodstream. But then what you have are white blood cells, WBCs. They will eat this pathogen. But if you see, there are only 4 or 5 WBCs, so they will not be enough for you to win the game level. You will ultimately lose it. So you will have to basically kill the pathogen by antibiotics. The other thing that you might notice here is this bar that was not in the first 2 levels. This is what we call as a resistance bar. So if you plant more antibiotics, this bar will keep on increasing. And more the bar goes higher, your chances of winning the game level go lower, because now you are generating bacterial pathogens which are very, very resistant to antibiotics. So you will have antibiotics, but they will not work on the pathogen. Okay. So I'll show you quickly how the game interfaces one more time. So this is how you start planting antibiotics, and this is where the pathogen starts to come in and you kill them. And when the bacteria starts to divide, then you need to make sure that you are planting antibiotics quickly, because if they divide, you are going to get half the marks. So every division is going to cost you score. So this is how we have built in a penalty for you to act slowly. And then comes the resistance bar. So if you see, as and when I am planting the antibiotics, this bar is moving slowly and it will keep on moving slowly. And what you realize is at some point, the bar will start to move very quickly. Right now, you can barely see it moving, right? But as and when I keep planting more antibiotics, it automatically suddenly takes a lot of speed and it will pace up. And there is a reason behind that. The reason is that resistance increases in a sigmoidal curve, and we'll see what it means very, very soon. And this is something that one has to avoid, okay? So game assets in Armor are based on scientific concepts. We don't want the players to get bored with a lot of education content in the game, because then you will not really like to play it. You're already reading a lot in your classroom settings. But the whole objective is also to make sure that the game is based on sound scientific concepts. So when the bar, the resistance bar on the top of the game is moving, it is not moving randomly. There is a sigmoidal function behind it. So when it will move initially, as you can see here, a new variant. Now we know SARS-CoV-2 variants, right? Looks likewise, when new bacterial variants come, it will take some time for it to establish, and all of a sudden its frequency in the population increases. So the resistance increases in a sigmoid, and this is what is actually built into the game. And the message is, if the player uses more than required antibiotics, higher bacterial resistance is going to be seen and the player is not going to win the game level. And this, I think, is a very important message. Less antibiotic, you are not winning. More antibiotic, you are not winning. You need to be at an optimal antibiotic dose to win the different game levels. As of now, we have in all 6 game levels for the skin infections and 2 for lung infections. One of the lung infections we are showing is tuberculosis, and the other one, of course, is COVID-19. The reason of introducing so many levels is because every level conveys a message. So that is why it's important for you guys to now come and enjoy and play this game so that you learn better in terms of what's really going on, why some antibiotics don't work on some pathogens, and how different antibiotics work in different fashion. So you can actually explore all those concepts in these levels. Now the lung level was introduced to basically demonstrate combination antibiotics. Now, if to a TB patient you give a single antibiotic, nothing is going to happen. The patient will very immediately convert into a multiple drug resistant patient. So even to treat a drug sensitive TB patient, you give 4 antibiotics. So we wanted to demonstrate this, that sometimes you need a combination antibiotic, and if not given, then the infection is not going away and you're going to keep losing the game level. The COVID-19 level was introduced primarily to very strongly convey the message that antibiotics don't act on viruses. It's only our immune system as of now that can address the infection. So as I told you, as of now, Armor has 2 worlds, skin and lung. We are working to add other worlds as well. Skin has 6 levels and each level has 1 learning message. But the overall message put together of all the worlds and all the levels is that do not underdose or overdose antibiotics, because both of these lead to drug-resistant bacteria. And what we can witness in very near future is that even a mild wound that gets infected can prove fatal. And this is a very much a reality now, because there are totally drug-resistant pathogens in the world right now. So it's not something that we're talking futuristic, it's already happening. So we need to act on this right away. And the problem is, despite the fact that COVID, we all know about the COVID-19 pandemic, we know the cause of the pandemic was a virus, we know antibiotics don't work on viruses, despite the fact that a lot of antibiotic prescription has happened during the pandemic, and it has basically impacted the deaths because of AMR. So it has been severely hampered. Imagine we don't even have a real time or accurate data of what has happened in past 2 years. So yeah, definitely COVID-19 has impacted AMR in a very negative way. The positive aspect of COVID-19 is, since now a lot of technology is being applied for addressing COVID-19, the hope is now that we can apply or basically extend the same technology measures to address AMR. So we should actually take the best out of the pandemic and apply it for the benefit of the society. Now coming back to the game, now each level when you play the game, what you will see is an introduction screen, which will be the challenge that you will have to solve. Like say for example, in one of the screens, it will tell you that can you help Alex take full course of antibiotics. So this is a challenge, and now the game mechanics are designed in such a way that it will see how you address it. Now, if you lose the game level, there is a losing screen which will tell you why you lost. But even if you win the level, you will also get a winning message so that you understand what you did right, so that you have won that game level. And I think this becomes very, very important, because it's not just gaming, this is also reinforcement learning, where you are trying to learn what is the right behavior and what is the wrong behavior. Right behavior avoided. For a wrong behavior, it is not a penalty, but if you can go back and redo it and do a better job next time, you will not have the same problem. So this is where the game is as of now. So far, what we have been able to integrate in the game is How do you go do optimal dosage? We know bacterial division can be very, very dangerous, so you need to act early. There is an antibiotic pathogen mismatch. Not all antibiotics can work on any type of bacteria. Not all antibiotics work in the same fashion. They work in very different ways. And then when you prescribe less or more antibiotics, then bacteria evolve to more resistant forms. So these 5 messages are very clearly conveyed in the different levels. Now we are working on some other very difficult concepts to convey within the game. And this is something which is currently under development. Now, before we go forward, let us watch a teaser for the game, which actually conveys a lot of information on what the game is about. So just enjoy the video and then we'll resume our discussion. So as you can see, it's a team effort. There are a lot of people involved in game design and game development. So moving forward, what we are also doing right now is, if you saw on the bottom panel, you could see 2 types of antibiotics. When you cross level 5, you will see 3, and as and when you keep going forward, you will actually unlock more and more antibiotics. Now there is an AWARE classification that is developed by the World Health Organization, WHO, which is access, watch and reserve group of antibiotics. Access group is something it is okay. These should be the first or second choice of antibiotics. Watch group is only indicated for specific limited use and specific infections. And the reserve group is a last resort. It is only when there is a life-threatening infection. So the point is, we need to make sure that this concept, not by antibiotic name, but the concept of access, something that you may use right now, something that you should be careful using, like in terms of antibiotics, and something that you should not use at all. If it is some, if you have an alternative antibiotic in the watch or the access group, then one should not touch the reserve group antibiotics, because if they don't work, there is absolutely nothing else to treat infections. So what we have now doing is, in initial game levels, one should not be able to unlock antibiotics in the watch and reserve categories. It is only when a player accumulates points in the earlier levels of the game by assigning right antibiotic for the right pathogen combination is when you will be able to unlock new antibiotics. So this is something that we are working on right now. If you have more ideas in terms of how can we do this better, please do write to us. So far, we have basically done something which is called a playtest in France and India with a variety of age groups. And we got a very positive response on how the game is, how the game has helped the student community understand. We received a tremendous amount of feedback in terms of how do we simplify the game, how do we increase the replay appeal so that you would like to play it again and again. We have worked intensively on the game graphics, the content, and we are obviously working on more game levels. So you will have more things to come back and, explore more worlds. I'm also actually from my team's behalf happy to share with you that we were finalists in the International Educational Games competition last year. Now, this is something which is very huge because this was the first game that we developed. And it was a very good thing because, see, the whole point of developing Armor was to generate awareness. So when we could generate awareness at an international scale, it is, I think, an important, it made us happy because at least it is able to do what it is supposed to be delivering. But it's not the end. As the game goes on, we are working to develop several new levels and worlds, and you can help. So we are open to suggestions from all of you. And that's not all. The suggestions, if you give, actually come with a reward. So what is a reward? If you have an idea of how one can design a game level, then we actually will develop that game level and integrate that into the game. And that game level will be named after you. So then that becomes a part of the game forever. So if you have any such suggestions, please write to us at this email ID, gameon.armor@gmail.com. And there is no deadline. There is no limit to how many ideas you can bring. There is no limit of participation. Anybody can write. And so this is basically something that we really would like you to think about the problem of antimicrobial resistance. Have a discussion with your community, with your parents. Think about what is the problem that you think that, if you have simplified that concept for them, it would have been easier to convey the message. So think about those things and get back to us. We do look forward to your suggestions and recommendations. But let's do one thing today at least. Let's take a pledge. Let's take a pledge to, to basically take antibiotics as the doctor prescribes, not less, not more. Always consult a physician before taking any medication. Don't take antibiotics for viral infection, again, on the recommendation of your clinician. Don't take antibiotics prescribed to someone else, even if it is a family member, because the infection can vary. The dosage for you can vary. There can be other variations. Again, coming on to the point, please consult a physician before you take medicines. And at the end of the day, if you can prevent infection by eating healthy food, maintain good personal and environmental hygiene, that is something that will allow all of us to remain healthy. Because it's a community behavior. Last but not the least, please bring awareness to your community. So if you take a pledge that I'll bring awareness to my community on AMR, then this is also the theme of the World Antimicrobial Awareness Week this year, preventing antimicrobial resistance together. So be a part of this huge initiative, because it's very, very important that we realize this and we make other people realize about the intensity of the problem. Now, antimicrobial resistance is referred to as a silent pandemic, even in my talk title, right? The reason we call it as silent is because despite the fact the damage it causes, it is not discussed. It is not discussed the way we are discussing COVID-19. And that is a part of the problem. It should not be a silent pandemic. It should be something which should be discussed very openly. And it should probably be named as a catastrophic pandemic, because we are sitting on really a time bomb. And the day it really blows, it's not just infections. Talk of diabetic patients, talk of healthcare system, talk of surgeries, transplantation, everything will stop. So it's not just infections. Wherever antibiotics are prescribed, all of that will come to a complete halt. So the request is, don't keep quiet. And that is what we are not doing. We are not keeping quiet about the matter. We are raising awareness in all possible ways we can. And that is why what we are also have launched is something which is called as an animation series on antimicrobial resistance. And this will be a series of animations where we will explain concepts in very simple language for all levels of community, whether it is school students, whether it is somebody who is not from a healthcare background. I'll not show you the animation series now. You can go back to the CSIR Jigyasa virtual laboratory. So we are starting by, how the microbes were discovered. So obviously you needed a microscope. So how Anton van Leeuwenhoek discovered the Ferd Mamadou, actually invented the first microscope. How does it work? How were the first infections of pathogenic bacteria like Bacillus anthracis discovered by Dr. Robert Koch? What exactly did he do? And there are several such stories in our animation series on AMR. And then we are also developing one on what exactly Alexander Fleming did and what is happening afterwards. It is important for us to keep raising these issues again and again in all platforms so that we can address it. Now, all the link to the game, link to videos, everything is available on the Jigyasa platform that you can access via this URL. And again, coming back to the fact, in addition to the Armor team who developed the game and did the graphics, I have a very young dynamic student in the lab, Romita. She's our science outreach expert and a playtest moderator. So she's the one who is building these animation series right now. Now, one thing that I would really like to share with all of you is that it's not that the world and the countries are not realizing the importance of the problem. They are, and that is why in the World Health Assembly in 2015, antimicrobial resistance was discussed, and there was a global action plan that was put in place to address it. Now, one of the first objectives of the program was to improve awareness and understanding of AMR. That to some extent is going on. We are doing that because when you understand something, you can learn it better, and that can influence behavior change. But if you see, awareness is not the only thing that is required to address AMR. You need to develop new diagnostics, need to develop new drugs. There has to be better ways of surveillance of infection. How do we optimize antibiotic use? How do we reduce infections? So collectively, this will address AMR. And that is where our laboratory is not just working on AMR awareness. For a very long time, we have been working to understand disease biology, what happens in pathogenesis, and how do we develop new methods for diagnosis and treatment. I'll not obviously get into details of this. You can actually look at this on our lab website. But what I would really like to convey to the students out there is that we work with a very, very large young research community through virtual collaborations. And they have immensely contributed to all the projects, all the infectious disease projects in the lab. Two of particular mention are school kids of your age. When they came to my lab, they were just finishing school. And they both actually contributed to international research publications from the laboratory, which are very well cited. So Pooja actually went on to become the Gates Cambridge Fellow. Ashma went on to become the Gold Scholar Fellow, and she actually got Johns Hopkins University, with a full scholarship of $80,000. So it's the point is these kids, when they started doing real life science, they were your age. They were very, very young. So the message to all the students out there is no age to do science. It's the passion that describes what you will ultimately do and how do you want to contribute. So don't let your age or degrees make you think what you can or cannot do. You can do anything if you're willing to do that. So have an open mind. And what we continue to do as part of ATL CSIR collaboration. So if you see, this is a very recent event we did in one of the schools we adopted in Chandigarh. We went to the school, we called the school kids to our lab. We discussed microbial diversity. We discussed antimicrobial resistance. We conducted experiments with them to show if you don't wash your hands, what happens, what kind of bacteria grow, if you wash it with soap, if you wash it with sanitizer. Do you really see a difference in growth of bacteria on these plates? We actually discussed a lot of scientific concepts on drug discovery, basic science. So we do a lot of these activities because it's important to, for a real life experience and engagement, it's important that we do this. Now, obviously, I cannot do this alone. So in the playtest activity, we have a team. So team is Romita, there is Nisha, Abhineet, and Manpreet. They all are PhD students, and they are actually doing this as part of their societal project. So I think this is very, very encouraging. And we continue to do this, and we will continue to do this till the time, we are actually allowed to enter schools. Hopefully they are open after COVID. So there are other people in the lab that I would really like to acknowledge. I have a lab in India, and I had a lab for a few duration, for almost a year and a half in Paris. So these are my lifeline, because they are the people who keep you going and keep you happy, motivated about doing good work. So I really would like to say thank you to them before closing. And I would really like to thank Niti Aayog, the Atal Innovation Mission, CSIR and Jigyasa for organizing this Demystifying series, because it's important that science does not remain only in laboratories. It should be understandable by general public. Until the time that happens, this connection will not build up and will not be able to collectively solve any problem which actually impacts irrespective of whether you're sitting inside the lab or not. So I think this initiative is very, very crucial in that context. And in the end, I would like to close by saying that let's play and learn how we can defend ourselves from the scourge of antimicrobial resistance, because we know it's a problem that needs our immediate attention. I sincerely hope that I managed to convey that message across to you, and I would really look forward to your feedback on how Armor is and how we can again work together and make it better. With that, I would like to thank you. And
Thank you so much, ma'am. It was a really wonderful presentation. I think I have understood most of the part associated with AMR, and the game also looks so informative. I think even I would be trying to put my hands on and see that how well I can do in this game. Ma'am, a few questions I was wanting to ask you. Firstly, you have already mentioned that we can access this using Jigyasa website. Am I right? These games, how can we access this?
Yeah, so the games are available on the Jigyasa website under the activities window. So the videos, the games, all the other active, whatever we develop as Jigyasa content, we make it freely available online.
Okay. Ma'am, in general, I was wanting to ask that whenever most of the time in our normal days of sickness, we can't really differentiate between a microbial infection or a normal infection. So many a times, as you have mentioned, is very rightly that we tend to take some medicines without any prescription, like thinking we'll be fine by it. So how to avoid that, and so that we should not contribute in this antimicrobial resistance cycle? How to avoid that?
Right. So again, the point is most often similar. So the symptoms of viral and bacterial infection seem similar, right? And that is where the problem starts, and that is where you need to basically go and meet a clinician. See, a viral infection normally. It again depends. There are so many viruses and bacteria these days. It is very difficult to say how long it's going to last.
Yeah.
But the whole point is, see, one of the issues also is that even a clinician cannot take that decision just by looking at a patient. They may need to do a throat swab or take a sample and do culturing. Now, when you culture something, it doesn't come out. The results are not available to you in a matter of hours. It normally takes somewhere between 48 hours to 72 hours, which translates into 2 to 3 days. Now, if you are sick and coughing and having fever, you may want a quick solution to that. And it might as well be a viral infection. And you really don't need an antibiotics. If you basically keep yourself hydrated and sleep, it'll go away on its own in 3 or 4 or 5 days. But the problem is that what a clinician can do in this situation, we will have to give them tools to address it. And that is where what people are doing today these days is sequencing. And I think COVID-19 has shown that we can actually sequence at scale in a clinical setting, because we have been doing that across the world now. So I think this kind of a mindset where sequencing is becoming one of the first readouts to differentiate between viral and bacterial pathogen, that's the first problem. And the second problem is, what bacteria is it? Gram positive, gram negative, what kind of resistant it might already have. So you don't prescribe those antibiotics, you prescribe something else. So it is not only about identifying the microbe, it is also, we need to also identify if the microbe is the one which is resistant to A, B, and C antibiotics. So these three are not going to work. So you give them something else. Yeah. So it's, we need more sequencing based methods where you can sequence the DNA of these microbes and then use them for basically making the diagnosis. And this can happen in hours.
Yeah. Ma'am, you have mentioned about the mistakes we have committed during the COVID-19 pandemic. And as on the other hand, this monkeypox is also arising. So, do you have any suggestions, like not to repeat those mistakes what we have already did? Like, so we don't want to repeat that.
Right.
Any
See, the point
suggestions? Yeah.
was, see, in COVID-19 also, why we have to understand why clinicians were, why antibiotics were prescribed. So there were primarily two reasons. One is that there was a lot of confusion between the symptoms of COVID-19 or similar to something which can cause bacterial pneumonia. So the symptoms overlap. The second problem is doctors, in case of severe patients, were avoiding secondary infections. So it's not, I would not say it's their fault. It's just that only a few of them were careful enough to prescribe antibiotics. Everybody else does it blindly. In case of monkeypox, I think we may not do that mistake because it looks like if you are infected, the infection is going to show up because it is something which is very visible. In COVID-19, it was not the case. So if somebody is infected, there is no way to really look at it and say the person has COVID-19.
Yeah.
Hopefully, it may not be the case, and we just hope that monkeypox remains like a local outbreak and dies off, and not really spread anymore.
Yeah. Ma'am, another thing is that what type of research trends right now going in this field? As I am a researcher, so mostly we are working upon this. We are also working on antimicrobial resistance, but how do we work is trying to find some analogs of already present antibiotics, just trying to change the structure of the chemicals. So, can you just brief about what type of different trends which are right now going on in the field of research?
See, there are two dimensions I think which are basically very actively being worked upon. One is to the use of phages. Obviously, phage resistance is also something that you can see, but still, phages is something that people are trying to work on. The second, I think, is mostly it's either a peptide-based antibiotic or something that can actually do genome editing. So, there are these new techniques that people are now wanting to use. And we know peptide-based antibiotics are actually out there. And they work. So basically, one of the issues one has to remember is 99% of microbial life does not stay in planktonic forms. They stay as biofilms. They stay as a community.
Yeah.
Most of our drug discovery efforts are focused on pure cultures, on planktonic forms, and this is not the natural form in which these microbes survive.
Yeah.
One has to look at the natural forms in which these microbes survive best, and then make sure that any compound that we predict or that we identify as to be antimicrobial is also tested in different forms of how the infection establishes in actual site of infection.
Right.
So that will help you really bridge this gap between what you discover and will it actually work or not.
Yeah.
And in terms of chemical discovery, I think there is a huge opportunity. People have been able to map the chemical space that can be synthesized. So I think this is the time for people who do computational drug discovery to really, really identify new chemical spaces, because we know chemically it is possible to synthesize them and test them.
Yeah.
But since the chemical space it's huge. So the computational biologists or basically people who do cheminformatics can really find that space and shortlist a set of candidates that are really beneficial. The way AlphaFold has contributed to protein structures, a similar initiative can really help push the drug discovery frontiers forward.
Yes, ma'am. Absolutely. Thank you so much, ma'am. Thank you again for your time. It was really an insightful presentation. And this is all about today's webinar. To all the viewers, I would like to say that we will meet again with another webinar in the series with another topic, probably to provide an insight to you. So till the next time,